Can I start?
Yeah. Yes, you might.
Okay, great. Well, thank you so much, Sylvia. First of all, to give me a second chance, because the first time I completely failed, and it was my fault. Actually, the network on the campus was breakdown. I've never seen this before, and again, thank you for these kind words, and again for inviting me. I'm excited to be with you today to share a little bit of what we are doing in the lab and what drives us. Today, I'd like to take this opportunity to demonstrate how much the brain to trigger adaptive behavior not only have to integrate sensory information from the external world, but also from internal states in order to maintain homeostasis. In other words, I'd like to illustrate through this presentation and through various examples how the brain orchestrates adaptive behavior according to this coincident detection that the brain will make between internal and external signals. But before really starting, I'd like to present, oh, sorry. Well, sorry. Can you see my presentation?
We can see.
Yes. Is it working? Okay, yes, great.
Yes,
so first of all, I'd like to demonstrate, and I'm sure as it is in the case in your laboratory, that a lab has to be, as a brain, a coincident detectors in between friendships and talents. And of course, those two dimensions are not sufficient, but for sure they are necessary to drive research and go from failures to failures, but always with the same enthusiasm. So I would like to present during this presentation some work. Let me use a pointer. Done by Ilana Gavani, some work done by Gabrielle Pouzet, Mariana Alonso from Enzo, and Antoine Isson, and Enzo Peroni. So most of these works would come from these talented people. And first of all, I'd like to show you my visions, and I probably we could share and discuss further into this idea that the brain have to process several dimensions. And as I titled this slide, the brain have to multiplex 4 dimensions. Obviously, the first one is time. As you know, brain at time t and time delta t plus delta t is obviously not the same. Brain is a highly dynamic organ. But not only so, in order to understand the brain, one have to take this dimension about the development of a single brain, but also this history of evolution. So both are time, but again, the brain have to integrate signal from the external worlds. And from my presentation here, I will use these words of exposome to mean any signal coming from the external worlds. But the brain have to also integrate the third dimension about what I call here internal loans, signal from the internal states that will trigger behavior to maintain homeostasis. And finally, the brain, want to understand the brain, have to also integrate the notion of scales. All these internal and exposome signals have to be seen throughout different layers of organization, from molecules to behavior. Right, so let's see now how we should integrate those dimensions in my presentations. So in the first part, I'd like to mention some part of some pieces of work that we have done in this field of brain integrate sensory signals in order to trigger behavior. And I'm going to show you that if you change internal states, of course, like here in this case, stress hormones, for instance, those behavioral responses might be completely modified. So the example I would love to take is the old-fashioned, because we have been mainly working in this field for many years. And in these experiments, what we have found that the animals have been trained to recognize odorants cues. And when we challenge the animals by giving in the drinking water corticosteroids, in these cases, as you can see here, the sensitivity for odor detections was changes by twofold, the decrease of acuity of threshold detections. And not only this acuity was decreased, but the memory, again tested here about a month later after one trials, the memory was decreased. So here one example, very classical, we haven't discovered nothing here. This was very well known before. By changing internal states here, by giving corticosteroids to the animals, you change the level of perceptions. The questions came whether we have change in other kind of behavior, and we did. We challenged the animal to assess several behavior, and as you could expect, when you give corticosteroid to animals, you produce chronic stress, and you have the hallmark here of anxiety, depression-like behaviors. The shift to the right here shows you that for animal drinking corticosteroids, you have an increase in the durations for the latency to eat in this novelty suppressed feeding. You have also a shift toward the right in the splash test. All mean that you increase the immobility of the animals. Animals are stressed and they don't really react to the exposure as they should do. And by discussing with one of my friends, Gérard Hebert, who is an expert in immunology, for him, giving corticosteroid to animals was not a matter of behavioral depression, but rather a matter of immunodepression. So then we decided to work together and to see whether this kind of challenging of the internal loom, whether we could have also other changes, and we look for the gut microbiota. And this graph on the left part here shows you that for the columns on the top of this label line, these are the control, and with giving corticosteroid to animals, these are the family of bacteria that were increased. And below this dotted line, you have the family of bacteria that were decreased. And this was done by talented postdocs, Elena Siopi and Grégoire Chevalier. Grégoire working with Gérard Hebert and Eleni Siopi in my lab. And so with this, we found that by giving corticosteroid, we could have a gut dysbiosis, meaning that maybe what we were seeing in this change of behavior was not really a matter of changing sensory perception, but maybe by triggering changes in the body. And so if this was true, then we wanted to check by transferring this dysbiosis to animals that have been stressed, those who are drinking corticosteroids, whether if we transfer transplant this gut microbiota into healthy animals, whether we could trigger depression. In other words, we wanted to challenge the idea that maybe depression could be considered as a communicable disease. In other words, we use animals, SPS mice animals that were taking corticosteroids or were subjected to this chronic mice stress, and having those depressed mice then transplant this microbiota to germ-free or antibiotic-treated mice and see by any change of their exposome, but just by changing this internalome, whether we could have the hallmark of depression, reduction of adult neurogenesis, change in the in blood serums, change in the microbiota, and change in behavior. And indeed, what we found, these are the animals that were stressed. So this unpredicted chronic mild stress showed that these orange columns, all animals were expressing the hallmark of behavior, increasing immobility, increasing the latency to growth. And now we are going to look at the behavior of animals that were not supposed to be stressed under this chronic mild stress, but just receiving this microbiota transplantation. And as you can see here, all animals receiving the stress microbiota turn to express also the behavioral depressed phenotypes, while the animal receiving the microbiota of the control were not showing any sign of depression. So this means for us that by just challenging the composition of the gut microbiota, we could trigger, after days, we could trigger the hallmark of depression. So we wanted to see whether by looking into the brain, we could have changes that we could expect, such as, for instance, adult neurogenesis. It's well known to be impaired under stress protocol. So we wanted to see whether by transferring the microbiota, we could observe a reduction of adult neurogenesis. And this is the kind of reduction of adult neurogenesis we used to see under this chronic mild stress. But again, we were observing the same reductions, quite to a less extent, but yet significant reduction of adult neurogenesis in the hippocampus for animals receiving this stress microbiota. Again, I repeat, but not receiving at all any stressful environments. So we wanted to look for more minimizing this observation by just trying reductional approaches by in order of not doing microbiota transplantation, but just use the serum of this stressed animal and see whether there was something in this serum that could mediate the depression. And we use neurospheres. We've been working on studying the adult neurogenesis in a petri dish. Either this petri dish was receiving controlled serums or serums from the transferred stress animals. And again, we have been able to mimic the hallmark of depression in the hippocampus, shrinkage of the dendrite arborizations, as well as a reduction of the quantity of newborn neurons in the petri dish. And so now we are seeing that we don't have to do microbiota transplantation. Just by looking in the serum, we should find some factors that are able to mediate these depressions. And indeed, by a metabolomic analysis, we found two factors: the amino acids, as you can see here in the blue, and the yellow factors are the lipid metabolism. were both impaired. Animals were like burning amino acids and lipids. For instance, if we look for tryptophan, because tryptophan, as you know, is a precursor for serotonin, we found that tryptophan was reduced, and the precursor of tryptophan, the 5-hydroxytryptophan that is going into the brain, was reduced, explaining why we could observe depression by doing this microbiota transplantation. And for the lipid story, we had companion papers. We found that there was a reduction of the endogenous cannabinoids, and the lack of these signals on the cannabinoid receptors in the hippocampus was the factors that was involved in these depressions. So those studies help us to really show that in this idea that there is a kind of quite vicious cycles or virtuous cycles between the brain and the gut. Here I show you how the psychological threats from external words can lead to change in stress hormone in the blood, could lead to change in the immune responses in the gut, whatever is going to involve anyway change in the gut microbiota composition. And this gut microbiota composition changes will trigger, as we have seen, change in the brain and change in the behavior. After all, we should not be so surprised that such a gut microbiota could impair or could affect our behavior when you consider that more than half of the cells in our body is made from microbial cells. So we wanted to go further into this analysis of this gut microbiota interplay with the brain functioning by looking directly also from signals that could come from the gut microbiota compositions. And in association with Gerard Bell again and Ivo Boneca, we have been working on looking this different interaction between this direct, interaction between the gut microbiota, sending signals from the gut compositions and those especially neuropeptide, the signals that are coming from the fragments of the wall of the bacteria. I will show you how they can be brought to the brain by the blood circulations, and will act in a precise brain area to trigger change in feeding and body temperature control. And more precisely, here, those signals coming from either gram-positive or gram-negative bacteria are coming from this neuropeptide, and those neuropeptides will act into the cell, into cytosolic receptors called NUT2. And as we know, and luckily we have a synthetic ligand called MDP. MDP can turn on the NUT2 receptors. NUT2 receptors were well known to be expressed by immune cells and in order to trigger pro-inflammatory responses. And it was also known the link between NUT2 and variants and chronic inflammation, like such as the Crohn's disease, for instance. So this is what we knew at the time, and so we wanted first to check whether NUT2 could be expressed into the brain. And by using light sheet microscope, Ilana was able to look in different parts of the brain. This is the midbrain, as you can see, lots of bright spots here looking for different nuclei. You have here this parietal muscle that is highly intensely labeled, meaning it's a region expressing NUT2 receptors, and as well as the thalamus. So and we look into more details who is expressing. After all, I told you immune cells were known to express NOT2 receptors, and by looking EBA1 and marker for the microglia, we indeed could find some label cells which are microglia. But the surprise came when using NUN for neuronal markers, we were able to see neurons that were expressing NOT2 receptors. So the questions came now, since we are seeing NOT2 receptors in the brain, whether the natural ligand could come into the brain, because after all, we know several examples in which we have receptors in the brain that are recognizing ligands that are not natural ligands in the periphery. So there is a kind of sometimes recycling building blocks like receptors, and in the brain, after all, showing not to express in the brain does not mean that neuropeptide will express, will activate those receptors. So first of all, we try to demonstrate that indeed this neuropeptide could go to the brain by the blood circulation, and that was done by using E. coli labeled radio-labeled E. coli, and by giving gavage, and then get different organs 24 hours after the gavage, and look in different organs, kidney, liver, brain, of course, blood. And as you can see here, you have a staining into the blood, and you have a staining also into the brain, meaning that this gut bacterial neuropeptide can, from the gut, go to the blood, and from the blood to the brain. So having shown that, now we have the ligand, we have the receptors, but we don't have any function. So the classical Claude Bernard experiments will be to understand the functions. You have to remove it and see what's going on. This is what was done by using knockout animals, in the whole brain, removing NOT2, and as you can see here, in females, aging after 6 months, those females without NOT2 gain weight, as you can see here. And when you look for the food consumptions, all means old and young, only old females after 6 months, they overeat. So this was one of the main results, where now we can see that these NOT2 receptors in the brain somewhere is able to regulate the appetite. So, but yet we haven't found where and how come. So the second step came when we looked for different part of the brain, as I said, with the light sheet microscopes. And if you look at the heat maps and the p-values, green means inhibitions with CFOS activity. So using CFOS as a proxy for neuronal activity, we were surprised to see a strong inhibitions in the arcuate nucleus, a place that is well known to control homeostasis, food, temperature, sexual behavior, social interactions, and so on and so forth. So we were able to demonstrate by in using this MDP, so this agonist for NOT2 receptors, that we were able to trigger brain changes, and in particular in this arcuate nucleus, a dramatic decrease of neuronal activity, green mean decrease, and you can see other part of the brain where we see an increase. So now we have a change of neuronal activity. We have seen by removing the NOT2 everywhere in the brain, we will see some changes in food consumption. We decided to be more precise and come back again and remove now NOT2 only in this part of the brain and not elsewhere. And for that, we use flux animals, NOT2 flux animals, and combine this with adenoviruses expressing CRIP, injected only in this arcuate nucleus, and now see whether we have changes, and we can control time, of course, and see whether we could mimic what we were able to observe in the general knockout. And as you can see here, after injecting the viruses, once you remove the NOT2 receptors progressively in all animals, then you see there is an increase of body weight and an increase that is turning to be more and more significant over time after injections. But the beauty of those experiments is that you can remove NOT2 receptors, for instance, when you have animals in germ-free context. For instance, when you give them antibiotics and there is no change, even though you have removed NOT2 receptors. And once you stop giving animals antibiotics, then you can see the activation of NOT2 that is translated in change in the body weight with food consumption increase in animals that have been removed only NOT2 in the arcuate nucleus and not in other part of the brain. So now we have a clear picture where NOT2 expressed in the hypothalamic neurons, some of the hypothalamic neurons, we have the way to control the food consumption. Yet we don't know how does it work in terms of physiology. For that, we developed fibers, optic fibers that we could insert directly into the arcuate nucleus, and again use adenoviruses, but not anymore to express the Cray, but GCAM6 in order to monitor the neuronal activity through calcium signalings. And what we were able to observe, calcium transients over time in the basal conditions, but once the animals has been giving food, you can see clearly a dramatic reduction of this neuronal activity, meaning that after food consumption, there is a kind of inhibitory signals into this arcuate nucleus. Now, let's see what's going on if we inject MDP control, which is not able to turn on NOT2 receptors. In this case, of course, these arcuate nuclei are able to fire activity. But now, if we give animals not food or nor this control MDP, but the real MDP to turn on NOT2 receptors, where we are now able to mimic, to phenocopy in some way the food consumption just by turning on those NOT2 receptors in the arcuate nucleus. So we wanted to look and understand with more causality rather than correlation what's going on when arcuate nuclei are facing NOT2 activations or MDP agonist. And for that, we use patchland recordings in the hypothalamic slice, and we use MDP into the pipette and let diffuse MDP or the control that is not able to turn on NOT2 receptors. And over time, of course, you have some rundown of the neuronal activity. But once you have MDP in the patch pipette, I should say this is the wholesale recordings, then you can see that there is a dramatic inhibitory activity, inhibition on this firing activity. So this is now an idea by which we believe that MDP is able to act on these neurons by reducing the firing excitability by changing some properties. We have been looking carefully either by changing probably potassium channels or sodium current. That should be done in a further experiment to explain this effect. So this is a summary. We have mice that are able to eat, and once they eat, you increase the density of neuropeptide that goes into the blood, and from the blood goes to the brain, and precisely in this arcuate nuclei, you have a strong inhibition as we were able to demonstrate, and this strong inhibition of this nuclei is sufficient to turn off the appetite, or it's a kind of satiety factors. All right, so to finish now, what I'd like to show you is the fact that I've been explaining to you how the psychological environments, this environmental cues promote stress, distress, trigger gut dysbiosis. In turn, gut dysbiosis through blood flow might change the activity of the brain. I told you, change in the amino acids, change in the lipids compositions. We have seen also that the blood flow can bring into the brain neuropeptide that will also activate or change the activity of different nuclei in the brain. But now what I'd like to show you is that there is another pathway, which is a neuronal one. And I would love to finish by showing you the link between the vagus nerve and the brain. Maybe I should come back again to this slide. I tried to skip, but maybe this is a way to show you that the work I've been describing to you on showing not to could really affect neuronal activity. After all, this should not be a surprise when you see that the change in the gut microbiome composition is also influencing during development, as we were able to demonstrate a few years ago, the microglia function. So this part of the work I've been talking about on NOT2 showing direct action of the gut microbiota onto the neuronal activity should be seen as well as a mirror image of action on the microglia as well. So finally, let's go briefly on this link between the gut and the brain through the vagus nerve. I should mention here this collaboration with a gastroenterologist, Arisokol, and Damien, who was a postdoc in the lab, and we have been working on this way we could see the brain control by gut microbiota by changing the vagus nerve activity. For that, I would just mention that we were able to do some gut microbiota transplant from first age mice to young mice or age mice, and then we're going to see from age human donors into young or age mice, and we're going to see that there is a component of aging that comes from the gut microbiota. For that, we are able to do the fecal microbiota transplantations, as I said, to young or age donors. So the blue columns here show young into young and old into young donors. And with this test, we are testing this short-term memory, 24 hours, and the animals should recognize or not the change in the cage. For instance, here, this is novelty. We have a novel object, and the animal will spend more time normally if he's recognizing this is a novelty in the cage, in the environments. And indeed, if the young animals receiving young microbiota, you have a score of the animals spending his time in trying to see what's going on as new in the cage. But and if the animals are older, they don't do this kind of exploration for novelty. And what you can see is that the young animal receiving old microbiota, you have a loss of memory. There is no recognition of novelty. And we went into more details in the brain to see whether we could observe a change in brain activity. And here we have this increase of C4s when animals are recognizing they are facing novelty in the cage. and for the animals that are receiving the old microbiota, old mice microbiota into young mice, you have a loss of this memory in a quite similar extent to the old mice. In other words, we are aging much faster young animals that are expressing some phenotypes that are described in the age mice. And we did the same experiments with young mice receiving microbiota either from young human donors or adult human donors. And what you can see here from 4 donors, human donors, we have if the donors are young, we have a memory that is quite similar to the young without any microbiota transfer, while you can see here for the transfer of the old human microbiota, you have in young animals a loss of memory. And we went into details to see what's going on in this transfer of information from the microbiota to the brain. And we look into the first relay into the brain, into the NTS, and using CFOS, what we were able to demonstrate that there was a change. So this is the CFOS activity, young into young, and old into young. So meaning that when we have this microbiota, old microbiota transfer into a young mice, we don't activate the CFOS, we don't activate neuronal activity into the NTS. I will skip that, and then we look to see whether we could restore the memory by alleviating this impairment of the lack of sensory inputs from the vagus nerve into the NTS by using different strategy for activating chemogenetics by using an increase of activity by injecting CNO, as you can see. So the red trace is a control case. Again, we are erasing the memory of young animal receiving old microbiota, but when we activate the sensory inputs into the NTS using CNO activity, then we restore the memory. So this is really to leave you with this idea that not only we should study the brain as the whole organs, and for many years, and we are all investigating how the different parts of the brain communicate each other. But really, what now there are more and more evidences about this brain being an orchestrator of different organs communicating together in order to trigger appropriate behavior according to the sensory inputs and different internal communications. But also, we should keep in mind that there is this idea of human beings being an holobiont, a symbiosis between our cells and the microbiota. And finally, I'd like to leave you with this idea that if you look the morphology in between a brain and the gut, I see quite similar structures. And the reason why I would love to leave you with this quote, a little bit from Descartes: I am a holobiont, so I am. Thank you very much for your attention, and I will be much more than happy to take questions.
Thank you so much, Pierre. This was a really nice talk and a great summary of your latest work. It's very impressive and very nice storytelling. So I would like to invite people to ask questions in the chat. and to remind everyone that after after this session, we will have a 2-minute break, and we will go to an extended Q&A session. But for now, yeah, let's start with a couple of questions. So, if you don't mind, I do have a question here. You know, about looking at the first and the second part, mainly, you know, how do you see, for example, with the injection of these murine peptides, how do you see this relation of this microbiota that you are studying? Are you studying the effect from the side of pathogenicity, or from as it being commensals, right? Because what you see is that, for example, you have a decrease in appetite when you have the infection, right? So I'm trying to think here if these bacteria that you are looking at are acting as commensals, or if they are pathogenics. And for example, have you looked into humans? Have you done any translational work and seen, for example, that major depression patients have increased in these microbes or not? How do you see it globally?
Right. I don't want to have a stronger reduced vision of human beings and depression that is much more complex than just a neuropeptide, of course. But your question is highly relevant when one have to decipher what is normal and what is pathology. Of course, I mean, by removing NOT2, for instance, we see that NOT2 in a healthy situations will be used as a kind of satiety signal, so not eating anymore. And as you mentioned, in the sickness behavior, well, we all know that there is a change in sleep, there is a change in food consumption, there is change in the body temperature, of course. So I think it's not a matter of changing the different signals, but it's more adding more complexity, because then you're going to recruit immune signals that are going to be not really linear, but just by bringing more and more other signals, you might change completely. So this idea of normal and pathology come from this idea you have a core signal that is used in the healthy cases. Here, for instance, I just try to make a brief summary of what we have been doing on NOT2 in the hypothalamus. But if you remember, you have seen a lot of staining of NOT2, for instance, in the striatum. And as many of you knows, striatum means reward circuits. And so maybe with food and reward, you might have something to do here, another case. But still, food is very appetitive. Maybe NOT2 activation there is normal, is healthy, but an excess, because then you might add other signals, then you turn into addictive, and then you turn into obesity and things like that. So for me, the normal and pathology is just a matter not of changing the gear, but it's a matter of adding such a little bit more complexity, adding signals. Just to end, because I know we have some question now coming. Regarding depressions, we are working with Gerard also in collaborations where we want to see how much signal coming from the immune response signals, immune system will act in different part of the brain, and what should be done nowadays is really you can't, as it has been done for many, many years, to reduce depression, to change in a very precise circuits. But it's again, depression, but other, as we were discussing about addictions, is more a failure of communication of systems, microbiota, immune, and neurosystems. So we should always now embrace this triad here.
I totally agree. Thank you very much for your very comprehensive answer. So yes, as you said, Pierre, so we have a few questions. I think I will read it out loud for you. So Azure Roll says, great talk, thank you. Does the microbiota go back to normal in the young mice after transplantation?
I must say, great questions. We have not been patient to look and pursue these experiments. What I've been told by people who are working with germ-free mice and then challenge, and there is a kind of memory of the signals. So whenever you are challenging the microbiota, usually after this gut microbiota transplantations, you of course change the microbiota, but it seems that the composition come back quite fast, actually, to the initial state, like if there will be a memory, a kind of place where there is a more stable equilibrium. So, but we haven't been looking carefully, and I think this should be very, very interesting. For instance, if you again challenge, you repeat those kind of gut dysbiosis, whether you might have a memory, what we used to call in mental disorders, neuroprogressions. You know, people who are facing stress and have episode of depression, if you repeat it, then you increase aging, for instance. You have like a memory, a trace that you have a cumulative effect. So, but we haven't been looking carefully whether we have we go back to the normal after transplantation.
So Benjamin Villasana asks, have you tried to cut the vagus nerve at the intestinal level? Thank you.
No, not at the intestinal level. No, that's a great question. And again, this should be done by yes. Further, I mean, this will be a follow-up story.
Absolutely. Sahil asks, do you think if the stress hormones can promote inflammation in the periphery or the brain? So I guess he's asking if the stress hormones are making more of the inflammation in periphery.
Stress hormones, when we are working on behavior, we have paradoxal effects. When you give corticosteroids to animals, before what I show you, inducing depressions and immobility, and you have a window in which you have, in fact, a mania. So you increase activity. So I don't know if this is a marker of changing inflammatory responses that might change over time by repeating, you know. So we know that corticosteroids produce immunodepressions, but also can activate other immune responses. But in terms of behavior, this is clear. Giving corticosteroids, initially animals are more active. So it's like a bipolar subject, actually.
So Rita Figueiredo asks, a great talk, thank you. Have you ever tried transferring microbiome from younger mice to older mice and see if it improves their behavior?
No. We did this in the past with parabiosis, but this is another story with changing the blood circulations. But we haven't done this with microbiota.
So and Regine Roer says, amazing talk. How does NTS activation leads to hippocampal activation and increased memory? Would other modalities of NTS activation work too?
Right. So I tried, but I encourage you to see the paper. It was published a few months ago. And we have actually a few days ago published another paper about depression and vagotomy. Yes, we are now, and we are working on the link between NTS and hippocampus, or NTS and different part of the brain, nebula and other part of the brain, to show that the story of depressions and hippocampus is quite simple. There are many other part of the brain that seems to be involved in depression that are hippocampus independent.
So Adelie says, thank you for the great talk. I would like to know if you performed any kind of comparative analysis between behavior performances of donor mice and recipient mice. Maybe as a control here. So, Adelie and Celine.
I don't get, sorry, the question, because we did use control mice. I mean, are we talking about on the aging effect here or on depression?
No. So maybe
because yeah. Go ahead. Sorry.
Depression. She answers.
Depression. Well, we had the control mice that were not stressed, of course. What was more important is really to demonstrate that just a serum by itself, either coming from animal receiving the stress microbiota, or serum coming from animal that receive controls microbiota, was enough to demonstrate that we were inhibiting neurogenesis. And then we found this lack of tryptophan to explain the reduction of neurogenesis and lack of also endocannabinoid signals.
Which was really a nice story. So I will ask you, okay, let's try to go through these in the next 3 minutes. So Ziv Ronay asks, is the immune system involved in the gut-brain link? You nicely demonstrated. Can the old to young phenotypes also in immuno-incompetent mice? So I guess, do you understand the question?
I guess so, because indeed in this aging study, we are demonstrating that the gut microbiota changes go through the vagus nerve and change the NTS activity and change the adult neurogenesis in hippocampus. But of course, there are probably also immune responses that are mediating brain activity changes as well. And maybe one way to challenge this would be to use immunocompetent to really see the part of neuronal activity mediated by vagus nerve versus immune responses, I guess. Yeah.
Yes, in the immuno-incompetent mice. Very well. So Kama Atekani, great talk, thank you. I'm curious to ask, one, why this regulatory axis of MDP-NOD2 is relevant for female? Do you think that that increase of microbiota in males can be detected by other ways? And second, just wanted to specify if I understood correctly, age microbiota is lack of potency to stimulate brain. Is it directly or via vagus nerve? Thank you. So I think the first question would be the more.
Yeah, to say to give some information about the first questions, we know that because that was one of our hypotheses, maybe in young females, the BBB was not permeable to NOT2, but we haven't found any changes in the quantity that we could find of label MDP in the brain. So there was no possibility of seeing any changes, because we know, I mean, I'm sure here people know that the BBB might change in the permeability over time during development. So that could be one of the questions, but we have to discard this. No, we believe that there are some effect, maybe some masking effect of sexual hormones, and by changing the status of the hormones of females, we could maybe see some changes of the sensitivity. So we did gross experiments by using ovarioptimism mice, and but that was not relevant. So that again, if I will have to bet, we haven't been able to look carefully, but if I will have to bet, I think there was some confounding or masking factors of sexual hormone. At least this is what the hypothesis we have to favor, because the other possibility have been discarded one after one.
I think that an interaction with the oestrus cycle could be playing something. Okay.
And that will be relevant to what maybe could be seen also for menopause in women. So I think there is here an avenue to look between interaction between sexual hormones, NOT2 receptors, and or microbiota in general, and some brain activity changes.
That was just a theme of our last talk, so that's from last week. So a last question from Harshmet, which is a bit more speculative. So I think it will be funny to finish in with this note. So this is some very interesting data. This question is purely speculative. Sensory neurons in the gut have been shown to mediate bacterial immunity. I am wondering if the gut sensory axis is connected to the NOT2 story.
Yeah, that is very speculative and very tempting to make such an hypothesis. Since NOT2 is highly expressed in the gut, NOT2 is highly expressed in the brain, of course, one could see a potential transfer of informations. Yet we haven't looked at, but it's a very nice way to conclude, actually. Between because it's really reinforcing this idea of a gut-brain axis, and the brain without gut, or the brain without any systems will be really poor.
Absolutely. So it's all about reading the body, so the needs of the body. So let's finish for today. This was a great talk today, and a great Q&A session. Let me thank also the attendees for all these very interesting questions, but and more importantly to you, Pierre, for your endless patience.
Thank you, Silvia, for your thank you.
So let me just remind you that next week we will have Judith Eisen talking to us about microbial modulation of zebrafish behavior and brain development. So a nice segue here. And that I will make a brief break now of 2 or 3 minutes, and we will be back at 8 past hour. Okay, so we just have a brief time to drink some water and so on, and we will have roughly a bit more 20-25 minutes more of questions and to have the chance to meet Professor Lledo a bit more closer. So see you soon. See you, Professor Lledo. See you soon. You can mute and close your camera if you want for the next 3 minutes. Okay.
All right.
See you.
See you.